So if someone doesn't mind helping you guys, I can pick the eraser. If someone please. Thank you. So with the steps of, just like I asked earlier, I might give you 4 or 5 proteins, put them in order, or 4 or 5 steps, put them in order. So please remember them, like what comes first in the P side, what comes next in the A side, etc. So remember that it always starts in the P and formamethionine comes in. It's brought by IF2, number 2. The next one comes into the A side, brought by EFTU, right? Don't worry about the role of IF1 and IF3. So EF2 followed, IF2, right, followed by EF2, if you will, TU. They link by peptidyl transferase, and then EFG translocates them. And the process repeats until you hit the stop codon. Release factors come in, everything falls apart. That's translation. Questions about Chapter 9 is 2 parts, mutations and a gene transfer. So let's quickly go through some major features of the first part, which is mutation. With mutation, be able to, again, differentiate, first of all, between the DNA level, right, and the protein level. Well, let's start, actually. We have point mutations, frame shift. Point mutations can be at the level of the DNA or at the level of protein. At the level of DNA, you have transition, transversion. At the level of the protein, you have missense, right, nonsense, neutral, silent, whatever. Actually, neutral is an example of missense. What are examples of transition? A to G, right? G to A, C to T, T to C, etc. Transversion, you're going from pyrimidine to purine. Missense, you change the amino acids. Silent, you don't. Nonsense, you create a stop codon. Frame shift, you're adding a base, you're removing a base. Forward in the wild type, reverse back to the wild type. Does that make sense? So true or false, a forward mutation always changes the genotype. A forward mutation always changes the genotype. True or false?
True.
Of course, that's the definition of mutation. It changes the genotype, right? Does it always change the phenotype?
No.
No, because you could have a silent mutation. The mutagens, very quickly be able, again, big, don't worry about examples here, know what they do. Let me give you an example, or break it down. Mutagens, right? Very quickly, chemical, physical, biological. Let's start with the easiest. Biologicals are transposons. Transposons create frameshift mutations. That's it. Physical, we have 2 types, right? We have ionizing and non-ionizing. What are examples of ionizing X-rays? What are non-ionizing UV light? What do? X-rays break the DNA. UV light creates pyrimidine dimers. That's it. Chemical, there's 3 types that we talked about. Base analogs, base modifiers, intercalating agents. Base analogs, base modifiers, don't worry about the examples. But both of them do what? Point mutations. Intercalating agents, frame shift. Questions about? That's mutations. The next part has to do with gene transfer. So we started with plasmids. Actually, be able to differentiate between the 5 types of plasmids. So first of all, gene transfer. Again, big ideas. There's transformation, transduction, and conjugation. With transformation, naked DNA. So be able to highlight the big ideas. Naked DNA. There's 2 types, right? We talked about artificial and natural. Natural, we talked about 2 types, Haemophilus influenzae and strep pneumoniae. This one is specific versus non-specific, etc. So again, be able to answer questions without going into details, transformosomes and sequences, etc. Is it specific? Is it non-specific? Does it break the DNA 2-stranded or 1-stranded, whatever. Artificial is E. coli, so that's easy enough. Transduction, I'll come back into in a minute. Conjugation, remember that's where we discussed plasmids. So please know the major features of the 5 types. They're not essential. They could be linear, they could be circular. They're found in eukaryotes. They could be episomes in and out. And conjugation, be able to highlight the difference between an F+, F-, HFR F-, F prime F-. So you can go back to the notes in previous week session rather than me going over in detail. But be able to answer questions like which transfers chromosomal DNA only. Actually, let's answer this one, then we'll go. We'll continue. Which of these transfers chromosomal DNA only? None of the above, right? All of them have to transfer plasmid DNA by definition. They might transfer chromosomal DNA in addition, but not only. But again, this is the big idea. You don't have to go into more details than this. Questions about okay. So, and again, we'll go a little bit longer than 3:15 for those who are interested. But let me now highlight the new material, which honestly is much more straightforward, I think, and hopefully you agree. But there's 2 big handouts. We have viruses, right, and then medical microbiology. Same thing. I'm not going to ask you sizes, like 1,000 genes or 1,000 nanometers or 40 nanometers. If I do ask about size, it'll be relative. Smallest virus, okay? Or which of these is the smaller virus? Largest viruses, right? What the largest viruses are the... So for example, let's do this real quick. 1, 2, 3, 4, 5. This could be a question. Parvo. Mimi. Pandora. HIV. Mama virus. Put these in order of size. This is the mama virus. Right? The smallest of these is Parvo. It's in the notes. What should be the next one? We did not go over it directly, but these 3 are the big ones, 2, 3, and 5. So the next one after that would be HIV, which is smaller than the Mimi, which is smaller than the Mama, which is smaller than Pandora. That's the largest virus right there. That's one of the smallest. So that would be one question right there. Other questions, what is, for example, with structure? So do know the virus structure, for sure. And then remember, with the virus structure, we have the genome, right? We have the capsid. These are always there. They have to have a genome, they have to have a capsid. But they do not necessarily have to have what? An envelope or accessory proteins. Envelope is obtained from the host, right? The capsid is made up of proteins. The genome could be double, single, could be segmented. Which has segmented genome? The flu virus, and then in fact, any concept really in these two handouts could be, but the concept of a segmented genome is important, right? So it could be literally anything. Well, any nucleic acid, let's put it this way. They cannot be proteins. Any nucleic acid. Segmented genomes, that's the one in parts. The flu virus has 8 single-stranded RNA. Protein envelope is picked up by the host from the host by the lipid through the phospholipid bilayer. These are examples like lysozyme here, reverse transcriptase, etc. So viral structure will have 4 or 5 questions. Viral replication, let me go over it quickly. Same thing, viral replication. One question is put these in order. Almost assuredly, I'll give you the 5 steps of the viral life cycle, put them in order. They absorb, they penetrate, right? They absorb, they penetrate. There's biosynthesis. They mature and release. Please know the steps in detail. Absorb. If we talked about a receptor, please know it. So for example, the male-specific viruses which target the F virus, right? HIV, which has 2 receptors, CD4 and CCR5. Lambda, which binds to the maltose. Penetrate, only the DNA usually comes in. If everything comes in, they have to uncoat. They have to separate the capsid from the genome. Biosynthesis, they make everything. First, they replicate the RNA, well, they transcribe the RNA. They make late, early, which are enzymes, late, which are structure. They mature, put everything together, and then they release. If it's naked, they lyse. If it's an envelope one, they puff. That's the one-night stand, if you will. That's the lytic cycle. The long-term relationship is the lysogenic. Be able to differentiate between a prophage and a lysogen, right? So, for example, if this is if this is the prophage right here, the whole thing is called the lysogen, right? Lysogenized cell. Transduction, generalized, specialized. Do the same thing. Generalized, specialized. Right. In fact, we'll summarize it into two minutes. The last one, the medical microbiology, is basically just definitions and terminology. There's nothing, to really go over in detail. But let me do quickly transduction. Specialized. Actually, we start with generalized. Generalized, specialized. The example we talked about was generalized errors in the lytic cycle. Specialized errors in the lysogenic cycle. Here we're dealing with lambda. You do have to know the insertion sites of lambda. Lambda inserts between the biotin and the galactose operon. So in both cases what happens, instead of packaging its normal DNA, it picks up viral DNA and in fact, in many cases, very little viral, mostly bacterial DNA. It infects another virus, another cell, and transfers the bacterial DNA with it. You don't have to know beyond the details that I just described. Prions, viroids, Just like the clicker question that I asked yesterday. Viroids are blank that do blank, right? Viroids are RNA that infect plants. Prions are proteins that infect animals. You have to know a little bit more detail. They are very small RNAs, right? They do not encode proteins. They can form double-stranded areas. That small differences can cause severe manifestations in the plant. Not, frankly, much more detail than that. Prions, be able to match a prion disease with its animal, right? Kuru, humans, chronic wasting disease, deer, etc. Be able to differentiate between the prion normal, prion abnormal, right? Prion normal has more alpha, prion abnormal has more beta. It's a normal protein, it misfolds. You can acquire, what are the examples, Creutzfeldt Jacob, etc. Again, do know, for example, what is NVCJD. Don't know the exact difference between NVCJD and... Oh yeah, that's right. So, right, we compared NVCJD to CJD in the notes, at least I did, with earlier onset, etc. You do not have to know the details of that slide. Just know that new variant CJD is the human form of mad cow disease. Don't know the exact differences between it and CJD. Questions about this? Frankly, the practice quiz, and there's many questions from this chapter, will give you an exact idea as to what to expect. The last portion, medical microbiology, is I think even more straightforward. Here, it's all about definitions and obviously organisms. But let me give you an example right from the start. So for example, you have to know that the difference between pathogenicity and virulence, right? One is quantitative, one is qualitative. You have to know the difference between a sign and a symptom, right? So for example, a rash is what? A rash is a sign because you can actually detect it, you can observe it, you can measure it. You will get a question, and I always give it on a final when I teach this course. I can't give it on a final if I teach another course, but it's the one where you have to put the 5 stages in order of a disease, right? Starting with the incubation period, prodrome, etc. The virulence factors, if it's there, I mean, the slides are not, it's not a huge handout relatively, but please know it well because you will get also questions about which organism produces what. So for example, the flesh-eating bacterium is Strep pyogenes because it produces a lot of hyaluronidase, right? What are ringworm infections caused by? What's toenail fungus caused by? A fungus which produces a lot of keratinase, right? Because it has to live on the toe, which is keratin largely. That table between endotoxins and exotoxins, as I mentioned in class yesterday, you will get a good number of questions. Which of these is characteristic of an endotoxin? Which of these is a characteristic of an exotoxin? Right? So be able to make word associations. You will get questions like, again, with the, especially at the end. So for example, let me give you the choices. These are toxins. Right. So for example, which one of these inhibits protein synthesis? It's mostly too early to answer this, but just to give you an example of the types of questions. This is a protein synthesis inhibitor, right? Which one of these, and there could be more than one, which one of these cause loss of fluid and electrolytes? It's the cholera toxin, right? Which one of these is a membrane disruptor? The alpha toxin of E. coli. Which one of these cause paralysis, botulinum, and tetanus? Be able to differentiate the two. Botulinum is flaccid paralysis, relax, right? Botox is the one that removes wrinkles, right, etc. Tetanus is the one that goes into a rigidity. It's a spasm that is essentially, it's a muscle spasm. It's spastic paralysis. Pathogenicity island, just remember these are where virulence agents could be really, I mean, be able to differentiate between fomite, vehicle, vector, right? So a vector is what? It's an animal that can transmit pathogens. True or false? True or false? True or false? True or false? A fomite is a vehicle. True or false? Fomite is a vehicle, right? Because a vehicle is any agent that transmits pathogens. A fomite is an inanimate object that transmits pathogens. So a fomite is a vehicle, but a vehicle is not necessarily a fomite, right? Isosceles versus triangle. All isosceles are triangles, but not all triangles are isosceles. Vectors, biological, mechanical, etc. Signs and symptoms, we've already talked about that. Really, the virulence factors. Make note cards, whatever you want to do, but please know them by the time Tuesday rolls around. So I'll stick around because I have another meet session. But if you guys have questions while they are setting up, good luck on this and all your finals. And